Now for a brief review of Chapter 5. Chapter 4 was a pretty short chapter. Chapter 5 is a pretty big chapter. So, let's review it. Now, on the first page or two of Chapter 5, page 69, it goes over an introduction to organic molecules of the body. Now, under Roman numeral 1 of the notes, we talk about polymer principles. We have 4 categories of biological macromolecules: carbohydrates, lipids, proteins, nucleic acids. In all 4 cases, they can have single units called monomers, multiple big molecules called polymers. On page 69, it describes dehydration synthesis and hydrolysis. Dehydration synthesis is the reaction to build polymers. Okay? The lab this week covers that. Hydrolysis is breaking polymers. So when you digest food and you're hydrolyzing it, you're breaking molecules down. Now, carbohydrates include sugars and starches. Now, monosaccharides are simple sugars. They're usually 6 carbons and they usually arrange in these hexagonal rings. Glucose, fructose, galactose are common monosaccharides. Glucose circulates in our blood, common in nature. Fructose, also common in nature, abundant in fruits. Galactose occurs as part of lactose. Now, when we digest food and ingest these monosaccharides, those that aren't glucose, your liver converts to glucose. Disaccharides are sugars made of 2 monosaccharides. Examples: sucrose, known as table sugar, maltose, which is a product of starch breakdown, and lactose found in milk. Now, when you eat these, your digestive enzymes break them, you absorb the monosaccharides, those that aren't glucose are converted to glucose, and it all ends up as glucose in your body. Polysaccharides are big molecules. Starch is a plant energy storage polysaccharide. So all starchy foods are plant foods. Glycogen is how we store carbohydrates in liver and in muscle. Now, the liver glycogen helps with blood sugar balance between meals. Cellulose is a plant structural polysaccharide. It is found in plant cell walls, fibers. We can't digest it, so it makes up the roughage in our diet. Chitin is also a structural polysaccharide, actually an aminopolysaccharide found in the cell walls of fungi and also in the exoskeletons of arthropod animals like insects. Lipids, Roman numeral 3, are a diverse group of organic compounds that are mostly hydrophobic. Now, the first type are triglycerides, fats and oils. A triglyceride is made of a glycerol and 3 fatty acids. Page 75. Now, all the different types of fats, polyunsaturated, monounsaturated, omega-3, trans, these are all based on fatty acid structure. Now, if the fatty acid is full of hydrogens, it's saturated. If it's missing hydrogens in one or more locations because of a double bond in the carbon chain, it is unsaturated. So if it's missing in one location, hydrogens, it's monounsaturated. If it's missing in multiple locations, it's polyunsaturated. I did go over in the notes how, generally speaking, saturated fats are solids at room temperature. Unsaturated fats are oils, except for the tropical oils like coconut oil and palm kernel oil, which are saturated. Another rule of thumb is that saturated fats have a better shelf life. Hydrogenated oils are taking unsaturated plant oils and making them more saturated to thicken them up. That's part of how margarine is made. I mentioned in the notes how that can generate increased saturated fat content, which is less healthy because of the risk of heart disease, but also trans fats. A trans fat is an unsaturated fat, but the double bond and the arrangement of the hydrogens is backwards of the normal situation, and therefore the body treats it differently. The next type of lipid on our list is a phospholipid. Phospholipids make up cell membranes. Page 76 and 77 show you a phospholipid. The phospholipid molecule has a phosphate group and 2 fatty acid tails. The phosphate end is hydrophilic and can mix with water. The tails are hydrophobic and are repelled by water. So if you put a bunch of these in water, they form a phospholipid bilayer just because of the hydrophobic-hydrophilic interactions. Top of page 77 shows you that. Another type of lipid is a steroid, also known as a sterol. Sterols are not made of fatty acids, they're made of fused carbon rings. Examples: cholesterol, vitamin D, sex hormones, anabolic steroids, adrenal cortex hormones. Roman numeral 4 are the proteins. Proteins are for structure and function of the body. All kinds of proteins exist in the body: collagen, hair, hemoglobin, enzymes, antibodies, albumin, clotting factors, most hormones. They're all proteins. Most of your body's made of water, much of the rest is made of protein. Proteins are made of amino acids. An amino acid is a molecule that has an amino group on one side, a carboxyl group on the other, and something in the middle, okay? Which I've denoted the letter R as a functional group. There's 20 kinds of amino acids based on what goes in that R position, okay? Now, the amino acids join end to end to form a chain of amino acids. Now, I spend a little bit of time on levels of protein structure. The primary structure of a protein. Oh, by the way, amino acids are shown on page 79. I am not making you memorize names of amino acids for lecture. The ones you built in lab, you need to know what those are. Page 82 shows you the levels of protein structure. Every protein has these 4 levels. The primary structure of a protein is the amino acid sequence of the protein. The secondary structure involves folding or coiling of the proteins caused by hydrogen bonds between amino acids in the chain. Then a tertiary structure involves more folding and coiling of the structure into a 3-dimensional polypeptide. Now this is caused by reactions of, or interactions of the R groups of the amino acids. And the quaternary structure involves multiple polypeptides joining into a functional protein. For example, hemoglobin is made of 4 subunits, 4 polypeptides. That's pages 82, 83. Now, proteins have a definite 3-dimensional structure. They have a definite shape. Shape determines function. Anything that affects the shape of a protein will affect the function of a protein. Extreme temperatures, extreme pHs can disrupt protein conformation and denature or ruin the protein. Now, nucleic acids, the Roman numeral 5, the 4th type of biological macromolecule, includes DNA, RNA, ATP. DNA is deoxyribonucleic acid, RNA is ribonucleic acid, ATP is adenosine triphosphate. Now, DNA and RNA are polymers of nucleotides. ATP is only made of 1. So these polymers of nucleotides are joined by the sugar and phosphate of each nucleotide. Now, we'll talk a little more about DNA structure in another chapter, but DNA is a double-stranded, double helix polymer. RNA is single-stranded. Now, the nucleotides of DNA include cytosine, thymine, adenine, and guanine. Now, in the DNA double helix, cytosine C will always bind with guanine G on the opposite strand. Likewise, A and T will also bind with each other. That's called the base pairing rule. We'll have a lot more to say about the base pairing rule as we proceed through other chapters. And that concludes our review of Chapter 5.